Hello everyone, welcome to the seventh lecture of our series for CUET PG and GAT-B. So in this lecture we will be talking about C4 cycle, and in the previous lecture we have talked about C3 cycle, and in the more previous lectures like we have talked about the light reaction and the dark reaction. Okay, so now since we have came this far, so in the next lecture that will be on CAM pathway, the another lecture will be on photorespiration, and at last we will be covering one topic of water potential, and then the series will for plant physiology that will end. Okay, now the thing is that in this whole series, like we have touched upon the most important topics for your upcoming examination, and I have solved a couple of questions in each lecture. You can see I solve a couple of questions. So in the next lecture, that is going to be very special because in the CAM pathway lecture, I will, because the topic is too short, so I will be solving all the PYQs till now, like for all the topics that we have covered. Okay, so do attend the next lecture for the practice session, like it is must for you. Many of your concepts will be clear there. Okay. So in this lecture, we will be talking about C4 cycle, that is also called as Hatch and Slack pathway. Okay. So this C4 cycle, this is like we will be like covering this in detail today. Okay, so my name is Deepesh Joshi and I have qualified all these examinations you can see, and mainly my lectures are focused on CUET PG and GAT-B for this particular series. Okay, and another thing is that we will be touching on each of the concept in detail. Okay, so stay tuned. Okay, so let's begin with the lecture. So now you can ask me that, sir, we had a C3 cycle, then why do we need C4 cycle? What's the need of C4 cycle? So the need of C4 cycle, okay, it is due to a phenomena that is called as photorespiration. So what is photorespiration? That is the question. What is photorespiration? So photorespiration is a phenomena which is seen in plants. So as I told you that this Rubisco enzyme, this consists of both the activities, that it can bind to CO2 also and act as a carboxylase, and it can also bind to oxygen and it can act as an oxygenase. Since this has both the activities, and in plants like from the PS2, okay, there is a release of what do we call as oxygen, and because this oxygen is being released in the plants, basically what happens is this oxygen, it can react with this Rubisco and it can lead to formation of 2-PGA. That is a product, okay, and it reduces the efficiency of carbon dioxide fixation. So that is the major problem with photorespiration, okay, that this oxygen evolution, it can lead to the enzyme, like the utilization of enzyme for not only for CO2 but for oxygen also, because of which the plant, it is not able to fix the CO2 by like applying those with those particular resources, the efficiency of CO2 fixation, that is not 100%, okay, because of this photorespiration. So to tackle the problem of photorespiration, okay, the plants, they like evolved themselves into some adaptations, and those adaptations, the plants which have, so basically those plants, they were called as C4 plants because they have adapted themselves for to like tackle this problem of photorespiration, okay, and to carry out a new cycle that is also called as C4 cycle. So C4 cycle is not a new cycle. The ultimate dark reaction that is happening is by C3 cycle only. Only the location of C3 cycle and the way this C3 cycle, the CO2 will be delivered to the Rubisco for this cycle, that is changing. Rest everything remains the same. That is the most important point or thing you can think of. Okay. So this is one of the thing, and another thing that I want to tell you is, so basically for this C3 cycle there were some important advancements. Okay, so this was the anatomical. Okay, so anatomical adaptations. One was Kranz anatomy, and the another one was biochemical adaptation. Okay, of the C4 pathway. So in case of biochemical adaptation, you can say that it is a result of convergent evolution. So what is convergent evolution? When you are like performing the same function but with different origins or with different mechanisms, then that is called as convergent evolution. So here there is a biochemical adaptation. That means that there are new enzymes, new products being formed from their substrates, and those products are now being utilized for this cycle. Now you talk about Kranz anatomy. What is Kranz anatomy? So the word Kranz, it refers to, you can imagine it as a necklace. Okay, of small beads or something, or basically a necklace kind of thing. So the arrangement of cells in the form of this necklace kind of thing, okay, that has led to Kranz anatomy. And which cells? Basically, these are your bundle sheath cells, which shows this Kranz anatomy. Okay, so this is one of the important concept. And mainly, if you talk about C4 plants, so you can have different examples. You can have sugarcane, maize, okay, sorghum, rice, wheat, barley, okay. So basically, rice, wheat, barley, these are the C4 plants, but live as C3 plants, okay. And yeah, euphoria, that is your and all the other ones. So you have these kinds of plants which shows your C4 cycle. Now if you come over to the Kranz anatomy part, so basically I told you that this is nothing but a necklace kind of arrangement of cells. So here you are having basically two kinds of things. One is your upper epidermis and the lower epidermis. Between the upper epidermis and the lower epidermis, you have these mesophyll cells, and then you have these bundle sheath cells. The mesophyll cells are these circular cells, and the bundle sheath cells are these cells. Now the thing is, the mesophyll and the bundle sheath cells, they are connected via plasmodesmatal connections. Plasmodesmatal connections. So these are the connections by which these mesophyll cells and bundle sheath cells, they are connected to each other. Okay, so that transfer of molecules can happen. Okay, transfer of compounds, molecules, or like some of the important biochemicals that can happen because of these connections. Okay, now the mesophyll cell and the bundle sheath cell, what are the major differences? So the mesophyll cells, so if you imagine the whole of this scenario, if let's say these are your mesophyll cells and these are your bundle sheath cells. So this is mesophyll cell, this is bundle sheath cell. They both are connected through these plasmodesmatal connections. Okay. So now what is the case? In case of mesophyll cells, you are having the granule chloroplast. What do you mean by granule chloroplast, guys? So granule chloroplast are something which have both PS1 as well as PS2. This I taught you in the previous lectures. So if there is like the presence of both PS1 and PS2, then in this part mainly your oxygen evolution will happen because of the water splitting complex that is present in your PS2. Okay, so because of which your oxygen will be evolved. Okay, by splitting of water, and this oxygen is bad for our process of C3 cycle because Rubisco efficiency decreases. So now what is the solution? So if you think logically that Rubisco is something that is getting attracted towards oxygen, or it's getting like distracted, okay, from its aim. So that is the case of your students also. You also have to focus on your aim. You don't have to be distracted. So Rubisco is now getting distracted because of this oxygen. So it was focused earlier towards carbon dioxide, but now its focus has been shifted to oxygen, because if oxygen is there, it runs towards oxygen and it reacts with it. So now the solution is we have to separate both of these. We have to separate both of these like Rubisco and oxygen. So to separate them, to make the best solution is to make different compartments. And the one of the compartments, that is the bundle sheath cell that we have made, it is very important to make it impervious. So this bundle sheath cell layer that is impervious, okay, as it is impervious to oxygen or CO2, okay, or any gas. Now the gas cannot enter from outside. Now the only thing that you have to take care of is provide CO2 to this bundle sheath cell, so that Rubisco now it can become like, it can only be focused towards CO2. Okay, so how we are gonna do that? So we are gonna like react the CO2 with some compound, and once we reacted it with some compound, it will be stored in that compound, and then we can convert it into some other form, and then we can simply send it to the bundle sheet cell. Once that compound is like it reaches the bundle sheet cell, that compound can simply be like broken, or it can be decarboxylated to release the carbon dioxide, which can be accessed by the Rubisco sitting in the bundle sheet cell. And then again in the similar process like your what you call as the CO2, it can be now added to the RUBP. Okay, and once it is added to the RUBP in the presence of Rubisco, you can get your products. Okay, so the similar C3 cycle now can carry on. So in bundle sheet cell, basically your C3 cycle happens. Okay, that is the whole thing. So the bundle sheath cells, they also consist of chloroplasts, but these chloroplasts which are there, they have only PS1. That means they are agranular chloroplast or stromal chloroplast. Okay, so this is a very important point to note. So in mesophyll cells, stroma is less, bundle sheath cells, stroma is high, and the light reaction happens in mesophyll cell, oxygen evolution is there, in dark reaction is happening in the bundle sheath cell, and Rubisco is present in the bundle sheath cell. So this is one of the important concept that we have seen, and this is a diagrammatic representation of the Kranz anatomy. You can see that these cells, these are your mesophyll cells basically, which are there, and then this ring-like arrangement, this is something your bundle sheath cells are. And plant families having the C4 species, you can learn the families if you are giving CUET PG botany, because that is something that can be asked. So I have named them Asteraceae, and all those kinds of families are there. You can simply remember it. Okay. Now moving over to the next thing, that how does this C4 pathway actually functions? So I told you there should be something that should accept carbon dioxide and then form a product. That product should be converted into another product that is more suitable for decarboxylation, as well as like for storage of carbon dioxide and for transportation of carbon dioxide into the bundle sheath cells through the plasmodecimal connections which are connecting mesophyll cells with the bundle sheath cells. Now, what is the whole process that is happening? So before you understand the process, let me tell you one more important thing. The thing is that in case of your bundle sheath cells, okay, and in case of your mesophyll cells, you should understand that first is the concept of chloroplast. The number of chloroplasts are more in mesophyll cell and they are lesser in the sorry, the number of chloroplasts, they are more in bundle sheath cells and lesser in mesophyll cells. Then comes the concept of your like pervious membrane. So basically the bundle sheath cells, they are not pervious, but mesophyll cells, they have pervious membrane that can allow the exchange of gases basically. Then you can talk about the intercellular spaces between the cell. So if you talk about these bundle sheath cells, they are tightly packed, they have a very less intercellular spaces, whereas the mesophyll cells, they have good amount of intercellular spaces. Now the another thing that you have to understand is which enzyme is present at like in which kinds of cells. So basically, if you talk about, I told you that the dark reaction, basically the C3 cycle is happening at the bundle sheath cells, and that's why it will have Rubisco. Okay, and the mesophyll cell, it will have one enzyme that is adding the carbon dioxide to a particular substrate, that is a primary carbon dioxide acceptor, and converting it into a product. So that's something which is doing this, that is nothing but PEPcase. and that substrate, that is PEP, phosphoenol pyruvate, and this PEP case has only carboxylase activity. That is something that you have to remember, only carboxylase activity. Rubisco had both carboxylase and oxygenase activity. So this carbon dioxide can react with this PEP and it can lead to formation of oxaloacetic acid, and then it can have further changes. So if you see in this diagram, this flow chart of C4 pathway in detail, like you will come to know about the important things that are there. So basically, if we talk about the carbon dioxide in the presence of the enzyme carbonic anhydrase and water, this is being formed, forming the H2CO3, that is carbonic acid, and that can further use protons to basically form your bicarbonate ion. And once the bicarbonate ion is formed, that is the soluble form, and the carbon dioxide is stored in the bicarbonate ion, with the help of the peptase enzyme, PEP, which is your phosphoenol pyruvate, that is your 3-carbon compound, you can add the carbon dioxide to it, and once the carbon dioxide is added, it can lead to formation of oxaloacetic acid. So this is the important concept that you should be aware of. So you can ask me that, sir, how is everything happening? I can't understand. So I can tell you in a simple chemistry way. Okay. So you can see you have the phosphoenol pyruvate. Okay. Wait. Yeah. You have your phosphoenol pyruvate. Correct. This is your phosphoenol pyruvate. Okay. Now what will be happening over here is that there will be addition of the carbon dioxide, and there will be formation of this what you call as oxaloacetic acid. So with the help of this bicarbonate ion and with the help of this peptase enzyme, basically now what is happening is you are having the formation of an acid, that is your oxaloacetic acid. Okay, so that is being formed over here. Okay, once your oxaloacetic acid is being formed, this is 4 carbon, you can see, and this is 3 carbon compound, that is your PEP, right? So this is the thing. So you are getting a 4 carbon compound. Now what will happen is this oxaloacetate with the help of a dehydrogenase enzyme. So what is the function of dehydrogenase? Basically, dehydrogenase is something that is helping in the reduction process. So if reduction is happening, these are oxidoreductive reactions. Then NADPH must be oxidized to NADP, and the H plus that is released, that will help in the reduction, and because of this is a redox reaction, so this oxaloacetic acid, it will be further reduced to malate. So once it is reduced to malate with the help of a dehydrogenase, that is nothing but malate dehydrogenase. Okay, with the help of malate dehydrogenase, this will be reduced to malate. So if you get malate, what you will be getting CH2, then this O double bond, C double bond O will be reduced, and you will have OH over here, H over here, and then you will have CO negative. So this is your malate. Now once you get the malate, that is again a 4 carbon compound, with the help of the plasmodesmatal connections present between the mesophyll cell and the bundle sheath cell, the transport of malate will happen to the bundle sheath cell, and this bundle sheath cell has an impervious layer, so oxygen will never come. And once this is there, then so here you had a primary carboxylation where CO2 was added to PEP. Now here you will have secondary carboxylation because this malate will undergo decarboxylation first. It will release the CO2, and once it releases the CO2, it will convert it into pyruvate. Okay, now the malate with the help of a malic enzyme. So this is happening with the help of NADP malic enzyme. Okay, so here NADP is getting reduced, so this part is getting oxidized. Okay, and once this is getting oxidized, you from the malate that you had, you are able to like form pyruvate. Okay, so once you are forming pyruvate, what is pyruvate? Pyruvate is CH3, C double bond O, COO negative. So this is your pyruvate. So once you are forming this pyruvate, that is again a 3 carbon compound, you can see, because you are releasing CO2 in the process with the help of an ADP malic enzyme, and then this pyruvate with the help of PPDK. PPDK is an enzyme. Okay, that is a kinase basically. With the help of this dikinase, so this is a dikinase, so that will convert pyruvate back into phosphoenol pyruvate only. So this pyruvate that is formed over here, that will be again converted into phosphoenol pyruvate with the help of PPDK. Okay, so this is the whole thing. Now you can ask me, sir, that how many ATPs are being consumed in the process. So the answer is that to avoid the problem of photorespiration and to maintain a C4 cycle, you need to carry out, or you need at least two ATPs that needs to be utilized. You can ask, sir, but there is only one ATP hydrolysis. So let me tell you in this in detail. The ATP, it breaks into ADP plus PI, then again it breaks into AMP plus PI. So what is happening over here is that basically ATP, so this is the adenosine, you have three phosphates, right? Now, if this is alpha, this is beta, this is gamma. So if you break one high energy phosphodiester bond, you release some energy. But now if you are breaking two bonds, then the energy released equivalent to two ATPs. So that is the reason we say that here two ATPs are utilized, because the energy released is equivalent to the energy released in case of two ATPs. Because one ATP, when it forms ADP plus PI, then there is energy release, then that ADP forms AMP plus PI, then again there is energy release. So basically that energy release is equivalent to the 2 ATP hydrolysis energy. That is 2 ATP give rise to 2 ADP plus 2 PI, that is equivalent to that. So basically here you are getting the pyrophosphate and then you have the AMP. So that's why in the process 2 ATP are utilized. Then again this PEP, since it is now regenerated, this is the regeneration of phosphoenol pyruvate from pyruvic acid or pyruvate. Okay, so this is the thing, and the CO2 that was released from the malate to pyruvate conversion, that can be again undergo secondary carboxylation. It can be added to RUBP, so CO2 plus RUBP, and again you can simply form 3 PGA, and then the cycle will go on. That is the traditional C3 cycle that I taught you. So this is about the C4 pathway. This is about the location of enzymes. So the steps are primary carboxylation, that is the formation of bicarbonate, peptidase, and then there is a reduction, that is with the help of malate dehydrogenase for oxaloacetic acid to malate. How does the reduction happens? I told you this chemically also. Okay. Then there is a decarboxylation step, CO2 release, in which malate is getting converted into pyruvate, and the secondary carboxylation CO2 fixation happens with the help of Rubisco, and then there is a regeneration of PEP from pyruvate with the help of this dikinase PPDK enzyme. Now this is the standard NAD plus malic enzyme type. So in this yellow part, can you fill in the blank? So this is something that is unusual. So can you tell me what is everything over here? So from CO2 you generally form your bicarbonate ion. Then here pyruvate to phosphoenol pyruvate is something that you will be getting. Okay. And from this phosphoenol pyruvate, when the bicarbonate CO2 is added and everything is happening, you are getting oxaloacetic acid. This oxaloacetic acid is getting reduced. It is getting reduced because NADPH is getting oxidized. The enzyme will be malic dehydrogenase. With the help of this oxaloacetic acid, it will be converted into what? It will be converted into malate. Malate is the form in which CO2 is now stored. Now it will be transferred. And once it is transferred, this malate, again it will undergo decarboxylation. It will convert into pyruvate. Okay, with the help of NADP malic enzyme. And the CO2 can enter the C3 cycle. And this entire process, there is a what you call as a oxidation also, which is happening. Okay, because NADP is undergoing reduction, and then this pyruvate is again transported back into mesophyll cell, and then this ATP to AMP breakage is happening. Okay, and finally you are getting the phosphoenol pyruvate that is regenerated. So this is a type of C4 cycle. Depending on different C4 plants, they can have some variation in the C4 cycle. But the overall concept is same, that we want to store CO2 in some compound and transfer into bundle sheath cell so that Rubisco now can take the command and it is not distracted towards the oxygen. It is focused towards its goal of fixing CO2. Remember my concept. Now the PEP case, it can bind with all isotopic forms of CO2. Isotopic and normal forms, both it can bind to, but Rubisco can bind only with the normal form of CO2. So scientists, they tried for radio labeling experiments when they were using radio labeled carbon dioxide and they were reacting RUBP with it and then forming 3PG and everything. They were not able to observe any kind of product formation because like Rubisco, it can only bind with CO2, that is normal, that is a normal isotopic form of carbon, that is 12 carbon, C12 I mean. And if you talk about pepcase, then it can bind to all isotopic and normal forms of CO2. Binding affinity is high with CO2 and binding affinity is less than for the Rubisco binding affinity is less than pepcase, but the pepcase, it is only carboxylase, so its binding affinity for carbon dioxide is very high. But Rubisco is both carboxylase plus oxygenase, so its binding affinity is lesser as compared to pepcase. This is an important concept. So it can bind even if the CO2 concentration is less than 10 ppm, but Rubisco, it needs at least 50 ppm of CO2 concentration to bind to carbon dioxide. If the concentration is much lesser than 50, then and if oxygen concentration is reaching 50 or higher levels, then it will bind to oxygen. So Rubisco, it can play in both directions. It is the most distracted molecule, most distracted enzyme. It is like feeling that it can manage both the things, but if you are like getting onto two boats, then probably you will just part away. That is something you should remember. Now coming over to the another systems that can be there. So basically the second system of the C4 cycle, okay, NAD plus malic enzyme type. In this there are not much differences. Only thing is that when oxaloacetate is formed, it is converted to aspartate instead of malate. And then this aspartate is getting converted into oxaloacetate in the bundle sheath cell, and then that is again undergoing the processing. So the enzyme remains the same, PEP to oxaloacetate, that is PEP carboxylase. Oxaloacetate to aspartate, there is a difference. You have NAD plus malate dehydrogenase. Okay. Uh, wait. Sorry. Not NAD plus malate. It is aspartate aminotransferase, the enzyme that is carrying out this transfer. Then aspartate to oxaloacetate, again you have the same enzyme. Then alpha ketoglutarate to glutamate things are happening, and then here you are having NAD plus malate dehydrogenase that is leading to the oxidation of NADPH to NAD plus, okay, and the reduction of oxaloacetate to malate. Then this malate is now oxidized to pyruvate with the help of NAD plus malic enzyme, and then this pyruvate is converted to alanine with alanine aminotransferase. So whenever there is a change to amino acid, there is an enzyme called as aminotransferase that is responsible. Okay, and then you have the PEP carboxylase and all that. Okay, for your further things. So that is a thing. So this is one type. Another type, if I tell you, it is your phosphoenol pyruvate carboxykinase type, in which again from PEP to oxaloacetate you have PEP carboxylase, then to oxaloacetate to aspartate you have NAD plus, sorry, again you have aspartate aminotransferase, then aspartate to oxaloacetate, and then to phosphoenol pyruvate. So basically here what is happening is you are having this the things are almost similar, okay, and mostly like you are having both the kinds of things, okay. So you are converting the oxaloacetate into aspartate, and then you are converting everything into PEP. So that is like there are different variations, but the most important thing for your exam is this cycle, okay. These are just extra things, no need to remember that much, only you can remember the type of amino acids which are there. Okay, so I have summarized everything here. So you have the pepcase present in the cytosolic compartment. Phosphenol pyruvate to oxaloacetate acid is the reaction it carries out. Then NADP malate dehydrogenates. It helps in oxaloacetate acid to malate conversion. And then aspartate aminotransferate, I told you to aspartate it is forming. Then decarboxylating enzyme, NADP malic enzyme, NAD malic enzyme, phosphenol pyruvate carboxylase, carboxykinase. Okay, that is the thing, and they are carrying out these reaction malate to pyruvate, and that's all, and oxaloacetate to phosphenol pyruvate. This is happening. Alanine aminotransferate, pyruvate to alanine it is forming. Pyruvate phosphate dikinase that I told you, PPDK. This is the thing that is causing the regeneration, pyruvate to phosphenol pyruvate. And with ATP is being broken down to AMP, two bonds are broken. So the energy equivalent to 2 ATP is released. Okay, what I wrote. Instead of ATP, I wrote TAP. So it is ATP. And pyrophosphate, basically adenylate kinase, these all you know. So this is about that. And then we talk about the regulation of C4 pathway. So you can have NADP malate dehydrogenase thing, ferredoxin thyrodoxin system I taught you in the previous lectures. Pepcase and PBDK, they can be allosterically regulated by phosphorylations. How it is happening, let's see. So for pepcase inactivation, basically pepcase, it is inactive due to the presence of malate at its active site, so it is dephosphorylated and inactive. And there is the pepcase enzyme, that is a kinase that can add phosphate group. Okay, can add pi to your enzymes. So basically this is inactive, and when light falls, this light is causing the activation of pepcase kinase, and once it is active, it can lead to ATP hydrolysis and the transfer of phosphate, basically phosphorylation of pepcase. And once it is phosphorylated, pepcase becomes active, and once pepcase is active, then pep plus carbon dioxide, they will be simply able to form your oxaloacetic acid. Okay, so that is the whole concept behind this thing for the pepcase activation. So this is regulated by light intensity. Okay, you can see in the figure also, and this is the concept. You talk about PPDK activation. So this dikinase enzyme, basically it is regulated by light and all those other factors. So PPDK has an important histidine and threonine residue present on its active site, that is inactive in case of absence of phosphorylation. But as soon as your ATP that is being converted into AMP plus PPI, what is happening is that because of this phosphorylation event, mainly your histidine residue that is getting phosphorylated and it is becoming active, and once this is becoming active by this phosphorylation, you can again add a phosphate group at the threonine residue. Okay, and once that is done, it again become inactive. So it is activated by phosphorylation, and it can also be inactivated by phosphorylation, you can see. And once it is having like dephosphorylation events, then this threonine phosphate, when it is removed, it can be converted into active form. So it can be activated via dephosphorylation or phosphorylation or regulated by light, and inactivated via pyruvate also. So this is about the PPDK activation. Now coming over to questions. So we are done with the lecture. I have taught you C4 cycle in detail with structure also, the logic of C4 cycle, why it is necessary to avoid the problem of photorespiration, and all those things. So I hope this part is clear. Now we can solve some questions, guys. Okay, so questions, they are very important. So the first question that is there is, the C4 carbon cycle is CO2 concentrating mechanism evolved to reduce photorespiration. The following are stated as important feature of the C4 pathway. The leaves of the C4 plants have Kranz anatomy that distinguishes mesophyll cells and bundle sheath cells. Okay, so C4 plants, they have Kranz anatomy. Okay, and that distinguishes between your all these different kinds of things. So basically this is correct. In the peripheral mesophyll cells, atmospheric CO2 is fixed by phosphenol pyruvate carboxylase yielding a 4 carbon acid. Absolutely correct. In the inner layer of mesophyll, NAD malic enzyme decarboxylates 4 carbon acid and it releases CO2. So this is another thing. But does it happens in the inner layer of mesophyll cell? No, no, no. It happens in bundle sheath cell. So this is wrong. CO2 is again refixed through Calvin cycle in the bundle sheath cell. That is absolutely correct. So what are your correct options? Answers A, B, and D. That is your correct answer. Moving over to the next question, guys. The following statements regarding the effect of CO2 concentration on photosynthesis in plants. So, to solve this question, I have to tell you about a very important point, basically. Okay, so that is something which is important for this question and all those things. So, that is nothing but you can say the law of limiting factor. Okay, the law of limiting factor. What does this law states? This law tells you about the simple thing that if if some enzymatic phenomena or any phenomena is driven by more than one factors, then the rate of that phenomena happening is dependent on the most limiting factor that is present at the least amount, concentration, anything. So let's say if you say that photosynthesis, it is dependent on your light, it is dependent on CO2 concentration, it is dependent on temperature, it is dependent on water availability. So if I say you that if I give you a statement that what will be the scenario under low light conditions, okay? If I have low light condition and I increase CO2, then what will be the rate of photosynthesis in C3 and C4 plant? So this is a question that I can ask you. So basically, if you have low light condition, then this low light is a limiting factor. This is a limiting factor. Okay, and once it is a limiting factor, then what will happen is that if I am like with the help with light also, if you consider the rate with CO2 also, if you consider the rate with water or temperature also, sorry, temperature also you consider the rate, and with water also if you consider the rate of photosynthesis, okay, with water also, then what you will observe is that since light was the limiting factor, so if you are increasing light, then rate of photosynthesis will increase. But with increase in CO2 sorry, very sorry. But with increase in CO2 concentration, increase in temperature, and increase in water amount, the rate of photosynthesis does not change much. The reason is that the limiting factor is the one that is driving the things. Okay, so that is one thing. Another thing is about carbon dioxide. So basically here we are talking about the carbon dioxide. So if I talk about carbon dioxide, you should know that the atmospheric carbon dioxide is how much? It is around 0.038%. So you can call it to be around 380 ppm. That is the atmospheric carbon dioxide levels. Now in case of C3 and C4 plants, there is a difference. So for C3 plants, the threshold for carbon dioxide that is around 460 ppm, and for C4 plants the threshold is around 360 ppm. So what you understand from this part is that C3 plants, okay, already their threshold is much higher. So if you plot this in a graph, you will understand that for C3 plants, their threshold, if you consider with the CO2 concentration, okay, if you consider with that, you will observe that if this is for the C3, that it is getting the threshold like this at so no, this is C4 that is getting the threshold at 360 and this atmosphere is at 380. So this C3 plant that will be getting somewhat around your 460. That is for C3. Okay. This is for C4, this is atmospheric. So what you understand from this, that C3 plants like they are performing at the less than their maximum potential because their saturation level for carbon dioxide levels is much higher, that is 460 ppm. But C4 plants, their saturation comes at 360 and atmosphere has 380. So they are already in abundance with CO2. So now you read the question. The following are some statements regarding the effect of CO2 concentration on photosynthesis in plants. With elevated CO2 levels, C3 plants are much more responsive than C4 plants under well-watered conditions. So now you tell me, is this statement correct or is this statement incorrect? So the statement is absolutely correct. Why? Because C3 plants, they suffer from photorespiration and extra CO2, it basically suppresses your photorespiration and there is an increase in photosynthesis. And C4 plants, they already have your no, they don't have this problem of photorespiration. So C4 plants, they are much more benefited. Okay. So that is the reason that this is correct. In C3 plants, increasing intercellular CO2 partial pressure can stimulate photosynthesis only over a narrow range. So this is the second thing. Okay. So in C3 plants, if you increase the CO2, okay, so are you able to increase? Basically, that is the range is not narrow, it is a broad range because there is a good difference between 380 ppm and 460 ppm. Okay, so they respond over a broad range of carbon dioxide. So this is wrong. In C4 plants, CO2 compensation point is nearly zero. So basically, photorespiration is minimum in case of your C4 plants. Okay. So the thing is, if the photorespiration is minimum, so that is the reason that they require this much less amount of CO2 as they saturate at much lesser amount of CO2 at 360 ppm. So that is the reason that the CO2 compensation point is said to be as nearly zero. Okay, so that is the reason this is correct. Okay, the compensation point is nearly zero because they are already saturated with respect to the atmosphere. So the correct answer will be A and C. Okay, if this is clear, then next question. Quantum yield of photosynthetic carbon fixation in a C3 plant and C4 plant is studied as a function of leaf temperature. Following are some of the statements based on this study. So basically, if you see the statement A, that at lower temperature the quantum yield of C3 plants is lower than C4 plants. So now you have to understand one more important thing, that the photorespiration, it generally increases with temperature. Okay, so if you have higher temperature, your photorespiration chances will increase. Okay, so this is one thing. So now they are asking you that at lower temperature, basically the quantum yield of the C3 plant and all this. Okay, uh, yeah. So, if you see at lower temperature, quantum yield of C3 plant is lower than that of C4 plant. So basically, this statement is incorrect. Why this is incorrect? Because at low temperature, your photorespiration will be lower. Correct or not? Your photorespiration will be lower. And now C3 plants, like they work efficiently because now they will show lower photorespiration. Okay, and now they will have higher quantum yield. Okay, their quantum yield will be higher. Okay, and C4 plants, they will spend extra ATP. Now there is another concept that I have to tell you. So that is the concept of your bioenergetics. Okay, that is C3 and C4. C3 plants, they require 3 ATP, 2 NADPH for the fixation of carbon dioxide. C4 plants, on the other hand, they require the 3 ATP plus they require 2 ATP for the regeneration of phosphoenol pyruvate from pyruvate. Okay, so in total they require 5 ATP and the NADPH will be same for the C3 cycle. So this is for the C4 cycle. Okay, and this is same for the C3. So so this is something that is required to fix one CO2. For 6 CO2, what will be the case? That is to make one glucose, one molecule of glucose. Then you will be multiplying everything with 6. So it will be 18 ATP. this here will be 30 ATP, this here will be 12 NADPH, and here this will be 12 NADPH. Now you can see that C4 plants are spending 2 extra ATPs. Now come back to the question. So quantum yield means that, like, if you're spending lesser amount of ATP and you're generating more yield, you're generating more food, then that is better. So in that scenario, if lesser temperature is there, photorespiration is lesser. So the C3 plant, they will have a higher quantum yield because they are spending only 3 ATP, but the C4 plant, it is spending 5 ATP. So that is much as compared to the say for synthesizing the same glucose. So this is wrong. In C4 plant, quantum yield does not show temperature dependence. Okay, so basically C4 plants, they suppresses the photorespiration, so that's why quantum yield, it does not show any kind of temperature dependence there. Okay, it remains stable with the temperature change because, like, they are not affected by photorespiration, so a change in temperature does not change their quantum yield because their photorespiration is not changing, guys. So that is correct option. Since the photorespiration is low in C4 plant because of pseudo concentrating mechanism, quantum yield is not affected. Since the photorespiration is low in seafood plants, okay, the quantum yield is not affected. That is correct because photorespiration is low and CO2 concentration is due to the Kranz anatomy and everything. So basically, quantum yield is not affected. At higher temperature, quantum yield of C3 plant is lower than C4 plant. Absolutely correct. Okay, quantum yield at higher temperature, because if temperature will be high, photorespiration will be high. If photorespiration will be high, if that is high for C3 plant, then you will be losing your, like, you will be doing photorespiration, so you will be not able to synthesize the glucose efficiently and you will spend a lot of ATP, so that will be reduced in the C4 ones. So that is correct. So correct answer is B, C, D. Okay, D, C, D are correct. Coming over to the next question. Following are the certain statement regarding C2, C3, and C4 and CAM carbon metabolism in plants. Okay, so for the CAM pathway, I will tell all these things in detail. Okay, when I will be teaching you CAM pathway in the next lecture. For now, let's see in C3 cycle, one molecule of 3-PGA formed during carboxylation phase utilized for biosynthesis of sugars, fatty acids, and amino acids. So basically, not one molecule, generally it produces two molecules of 3-PGA that I already wrote in my previous lecture slides. Okay, and that is the thing. And they enter the reduction as well as your regeneration mechanism and not the biosynthesis part basically. So that is not that much correct option. For talking about during C2 cycle, glycine is transported from peroxisome to mitochondria and glycerate is transported from peroxisome to chloroplast. So I think this question I have to take in the next class because it is more for your C2 cycle and CAM pathway. So we can discuss it in the next class. So this question, guys, this is a GATB PYQ only. Okay. Tell me the answer. Which of the following combination of statements are true about C4 cycle? Malic acid formed in the chloroplast of mesophyll cell. Malic acid is nothing but malate. Okay, so if you are talking about acid, then that is a chemical species, and when it is malate, that means that it has lost an hydrogen, and now you can call it as ate. Okay, so that is same. So malate is, that is correct. It is formed in the chloroplast of mesophyll cells. Malic acid is transported to chloroplast of bundle sheath cell. Absolutely correct. Pyruvic acid producing mesophyll cell move to the chloroplast of bundle sheath cell. No, wrong. Reverse happens. Okay, malic acid is transported to chloroplast of mesophyll cells. Wrong. Reverse happens. So the answer is A and B. Answer is A and B. So thanks a lot for joining to this lecture. We have like covered C4 cycle in detail with questions, with the factors affecting photosynthesis also, that is covered in this question. Okay, all those things. So yeah, this is sufficient for your CUET PG guide. We remember the cycle. Remember the ATP expenditure, remember the differences in Kranz anatomy, and that is what is required. Yeah. So thank you for joining.